So. Ich hoffe, das ist jetzt auch auf Zoom sichtbar. Okay. Vielleicht. Einen größeren Mauszeiger. Then, okay, now welcome back. And I'm going to continue with this more introductory part of the lecture. What I didn't talk about last time was this kind of very brief overview of DNA-related kind of biochemical or enzymatic processes. These will play a role later on in the lecture. So I'm just briefly skipping through these next few slides before we will start with DNA thermodynamics, which is an important topic, of course. I also uploaded a tutorial sheet on DNA thermodynamics today, which we can talk about in the tutorial next week. I will probably also mention that later. So first of all, of course, DNA and the lecture is entitled Physics of Genes. And in a sense, this sort of course means that we are mainly dealing with DNA, but also with the genetic processes that sort of actually lead to the expression of genes, which means that essentially we talk about the steps that are involved in the central dogma, which, as probably everybody knows, that involves DNA being transcribed into RNA, and RNA is then translated into proteins. And there's also this replication step where DNA is sort of replicated into DNA. There's also this process of reverse transcription where RNA is sort of reverse transcribed into DNA. And all of these processes involve at least one or several molecular machines, in most cases enzymes. In this case, the ribosomes, of course, are kind of a mixture of RNA and protein. And all of these things have been studied in extreme detail, of course, over the last decades. And many of them also have been studied with biophysical methods, in particular single molecule tools. And we are going to talk about them in the course of the lecture, I guess. So here I just briefly mention that they exist. So in DNA replication, the fundamental problem, of course, is how to make two pieces of double-stranded DNA out of one double-stranded DNA. So in a very schematic image that looks like this here. So we have these two anti-parallel strands, 5' to 3' end direction here, and in the opposite direction, the upper strand. So these have to be somehow separated. And then one of the strands is copied into its complement. And that's why this type of replication is also called semi-conservative replication, because in the daughter molecules, you have one of the sort of original strands and one newly synthesized strand. So that means sort of half of the information is kind of carried over from the previous generation. And the production of these new strands of DNA, this is performed in a process called DNA polymerization, and the enzymes that are responsible for that are called DNA polymerases. So here the enzyme is actually not shown. The only thing one should probably remember is that the synthesis proceeds from 5' to 3' in the 5' to 3' direction, which means that if there is like a growing template, you will have a 3' OH group available here, and an incoming nucleotide triphosphate sort of is used to sort of make a new phosphodiester bond. So this pyrophosphate is split off in that process, and you have like an additional base pair, and the complementary base on the other strand, in a sense, tells you which base to incorporate. So we will talk about this process in detail later. It's also interesting from a biophysical point of view, because one of these kind of fundamental concepts in biophysics called kinetic proofreading plays a major role in this, to make sure that you really incorporate the complementary base and not one of the other three bases, sorry. So here, of course, we see this is performed by an enzyme. So this process actually doesn't work without enzyme, or at least it works extremely slowly. And that's one of the big actually issues in also the research on origins of life. So how such processes would occur when you don't have an enzyme. So that there's a lot of ideas about that, how to do this enzyme-free replication of DNA in this context. But it doesn't work under kind of conventional reaction conditions. So here, the enzyme DNA polymerase is often shown like a hand. It's a right hand somehow. So essentially this hand can sort of enclose the double strand, and the reactive center somewhere is kind of at this position, and this is where the end of this kind of single strand that is currently extended, or this strand that is extended, is placed. And we are also going to talk about this in more detail later. In the context of the double helix, like this process really is very complicated, because when you want to replicate a genome, this genome first needs to be unwound, and that creates this fork-like structure. It's called the replication fork. And now we have this problem that the polymerase only likes to polymerize in the 5' to 3' direction. Now, obviously, this works well on this strand, which is called leading strand, and it doesn't work well on the other strand, because the enzyme has to always start somewhere, maybe here, and then proceed in this direction. So if it wants to replicate maybe a sequence further downstream, you first have to wait until this is kind of, the double strand is unwound, and then it can start here. So what happens on this so-called lagging strand is that there are kind of consecutive sort of initiation processes, where first a primer is synthesized, and then the polymerase starts to synthesize DNA from there. And this very complicated process involves many enzymes. So first of all, there's a so-called helicase. Helicases are molecular machines, in a sense, that unwind DNA, and they require ATP to sort of open up the base pairs. And so that's, there are also kind of a lot of single molecule studies only on the helicase. So some of these enzymes actually have this kind of circular shape, or not circular, but ring-like shape, in order to keep them on the DNA track, so they cannot really dissociate easily, and that increases the so-called processivity. So in the language of molecular motors, a processive motor is a motor that runs along its track for a very long time without falling off. And so when you make a ring around like such a strand, you cannot fall off easily. So that's the one of the reasons why these look like this, probably. Then the other thing that you need for synthesizing DNA in the first place is, and sorry, I'm jumping back and forth, the polymerase doesn't start just by itself. It always needs to have this OH group at the 3' end of a short, of a DNA strand that is already bound to the complementary strand, and that's called a so-called primer. So you need something that generates the primer in the first place, and this is the so-called DNA primase. So there's an enzyme, or there are several enzymes that can act as primases. Also, RNA polymerases can prime DNA polymerization. So you need something to make a first kind of short stretch of RNA, and then the DNA polymerase can come in and start synthesizing from here. Then another thing that might play a role, and this, which is a problem, is that actually when you have single-stranded DNA rather than double-stranded, it might form extensive secondary structures. So it can fold back on itself, and this represents an obstacle for the enzymes that want to run through the want to kind of go through this sequence, and this can be so strong that the enzyme actually stalls. It cannot really continue to synthesize because it cannot break the base pairs in the secondary structure. So there are actually assisting proteins like this single strand binding protein that actually somehow make kind of flatten out, in a sense, the sequence. And now if you put everything together, you get this super complicated kind of replication fork structure, where the helicase first unwinds the DNA double helix. The DNA polymerase can synthesize easily on the leading strand. So this little sequence here, that's called the clamp, and this also improves the processivity of polymerization. There's another enzyme that's called clamp loader that helps to load the clamp onto the DNA. There's the single strand binding protein. Here is this kind of polymerase synthesizing in the sort of on the lagging strand, and the RNA primer shown here is actually produced by the primase. So there's actually, as a result, you will have a DNA RNA hybrid here. There's an additional enzyme called RNase H that specifically degrades RNA in RNA DNA duplexes. And then there is a gap in principle that can be filled with a polymerase, and then everything is ligated together with another enzyme called ligase. And that's shown here. So if you have a situation like this, where there's a 5' phosphate neighboring a 3' OH in the context of a double helix, a ligase can actually make a covalent bond here. And this is sort of happening at the end to sort of connect all the different fragments that have been generated on the lagging strand. So quite a number of enzymes is involved here. So this process actually, a colleague of mine, Karl Duderstadt, maybe you know him, he devotes his whole life only to this structure. So it's sufficiently complicated to work only on this. So he's studying essentially all the enzymes in a kind of purified form and does single molecule experiments on replication processes. So rather than just ligating things together or polymerizing, you also sometimes need enzymes to destroy DNA or to cut DNA. And there are different so-called endonucleases. And the endonucleases of type 2 are the ones that bind to a specific sequence on a DNA and cut in the neighborhood of the sequence. The type 1 endonucleases, they bind somewhere and they cut somewhere else. So here for type 2, they are kind of the cutting side is close to the binding side. And there are these kind of standard enzymes that cut like this. So they recognize that sequence here, and then they either make a so-called blunt end where they just cut both strands at the same position, or they cut at different positions, and this creates so-called sticky ends, because as soon as these two strands fall apart, they can rehybridize because they are obviously complementary in this region. And among these type 2, there are other types. There are subtypes called type 2, that's the standard, and type 2S, 2B, and so on. And type 2S enzymes play a role in certain cloning techniques. I'm not sure whether we are going to talk about this too much, but the THOC1 enzyme, for example, plays a role in Golden Gate gene assembly. And the advantage of these enzymes is that they actually cut at a different position than the recognition site. And this allows you to, in a sense, program the sticky ends that are generated by this process, because the sequence that is cut here is independent from the recognition sequence of the enzyme. So in principle, you can maybe cut somewhere and it will be like some freely chosen sequence. Nowadays, of course, people also do a lot of cutting with CRISPR systems, and this is something that we are going to talk about later in the lecture. And CRISPR enzymes like Cas9, they are in principle, they are so-called RNA-guided endonucleases, because an RNA tells them where to bind and cut rather than kind of these traditional enzymes. These traditional like endonucleases, they played an extremely important role in the development of genetic engineering. And briefly after the first isolation of the first endonucleases, people also invented cloning, because it was kind of clear to the people who invented that when you can create these sticky ends, then you can actually apply your endonuclease twice to maybe a carrier strand like this here, so-called a vector, if you want, and a DNA piece of your interest. And then by cutting, for example, this circular strand here and your DNA of interest, then these sticky ends will have the corresponding complementary sequence. So you can insert this fragment into your vector, and with this you can introduce pieces of DNA from some organism or synthetic DNA into another organism, and this is why it's called recombinant, because you can in principle mix gene sequences from different sources. And finally, I'm going to briefly mention this process called PCR. You probably also all know about that. That's a nice trick that actually uses DNA polymerases, and rather than using a helicase like in biological replication, you can just heat up your DNA strands and then they fall apart. And this is where actually thermodynamics plays also an important role, and things that we are going to talk about in the rest of the lecture are important for also understanding the kind of stability of these duplexes. So what is done in PCR is that you anneal short pieces of DNA to these separated strands, and they are called primer. And when you then add a DNA polymerase and the nucleotides to your mixture, you will just synthesize the complement of these separated strands. And then the main trick behind PCR is that you do this several times in a row. And that means that after the first round of replication, you separate these things again, you make another copy, and now the copies that have been made in the first round also act as templates for the next round, and so on. And that leads to an exponential amplification of your initial DNA that was present here. And therefore, this is, first of all, you can generate large amounts of DNA with that process. First of all, you can sort of select a certain piece of DNA, which is defined by the sequence, by the two primers, because they kind of flank the sequence that you amplify. And of course, because it's an amplification process, it is used for detection of DNA at very low concentrations. So this is why, of course, over the last years, everybody had to do these PCR tests with your corona tests, because the corona samples typically only contain very small amounts of DNA, and you needed to somehow amplify that, otherwise you cannot detect them. I think that in like these nose kind of swabs, you have very low concentrations on the order of attomolar, atto to femtomolar, and this is something that you simply cannot detect with an ordinary, let's say, fluorescence technique without pre-amplification. So you first need to amplify before you can detect that stuff. So that's this thing. And well, yeah, okay, that was only part of the central dogma. That was the replication part. We also need the production of RNA. And that's another process called transcription. So DNA is somehow converted into RNA by an enzyme called RNA polymerase. And again, this is somehow like a hand that grabs around a piece of DNA. And there, in contrast to DNA polymerization, you now have an intact double strand as a template. So one of the strands needs to be, again, the DNA needs to be melted here, and one strand is actually not going through the reactive center of the polymerase, and the other strand acts as the template for the production of the RNA, which is then going out through another channel in this protein. So I'm going through all of these processes in more detail in the rest of the lecture. That's just a very brief overview. So in DNA, RNA polymerization works like this, that in this case, that's E. coli polymerase. This polymerase needs to find a certain sequence on the DNA, which is called the promoter. In this case, the sigma factor actually binds to the promoter and recruits the rest of the polymerase. And this opens the so-called transcription bubble. And there will be the production of RNA. And at one point, the RNA might form some kind of major secondary structure which terminates the process. And the RNA is released, and the polymerase can synthesize another RNA strand. So these are these things. What I haven't, what I'm now talking about right now is the translation process, then which is much more complicated, and we are coming back to this later. Just one thing here that you cannot read this probably, but in the like maybe 10 or 15 years ago, people still thought that RNA mainly has the role of being either tRNA, a part of the ribosomes as a ribosomal RNA, or mRNA. But now it has become clear that RNA plays an extremely important role. And there are many, many different types of RNAs with all kinds of abbreviations, like snRNAs, no RNA, long non-coding RNA, microRNA, piRNA, and so on. And they mainly play a regulatory role. They somehow modulate gene expression processes, at least in eukaryotic systems. And it also turns out that, and that I think we talked about this in the last lecture, only a small fraction of the human genome codes for proteins. I think it's only about 2% or so. And all the rest of the genome codes for some type of RNA. So it's not that the rest of the genome is silent. Everything is transcribed, and it generates a lot of RNA, which is not coding for proteins. And this is something that has been become kind of obvious in the last years. And therefore, it's also important to study RNA, of course, as a part of this of the gene expression process itself. So that was what I didn't talk about last time, and I'm going to continue with the real with the topic of today, which is DNA and RNA thermodynamics, which probably will also take some time, maybe until next time. And I just stopped screen sharing because I was not sure whether this would still be visible in Zoom. So hopefully this will be visible. I again will just start with like preliminaries that are very general. So maybe it's not, I hope it's not too boring, but DNA and RNA are complicated molecules, of course. They contain kind of many different chemical groups, and therefore many different molecular interactions play a role in DNA thermodynamics. So I'm going to talk a little about the relevant molecular forces, but then they are not really important for us and for the rest of the lecture, just in case for people who really want to model, for example, DNA or RNA on an atomistic level, they need to sort of incorporate these things. Another thing is thermodynamics. So we are going to talk a lot about delta G's, like free energies and related concepts. And I wanted to give you a very brief crash course in statistical physics to kind of just introduce these topics. So this is just a recap for everybody who had kind of thermodynamics and statistical physics. So maybe you remember there are these thermodynamic potentials. And in statistical mechanics, we always start with U, that's the internal energy, and the natural variables for the internal energy are S, V, and N, because these are extensive quantities. So when you double the size of your system, then you get twice the amount, twice the volume, twice the entropy, twice the free number of particles, twice, and so on. You know what I mean? So the other parameters are these kind of intensive quantities that do not change when you change the system size, and that's something like pressure, for example, or temperature. And in many cases, experimentally, it's more convenient to use these intensive variables as control parameters. In particular, in chemistry, people like to keep temperature and the pressure constant, and therefore the quantity, this G, the Gibbs free energy, plays such an important role in this chemical and biochemical context. And because the description of thermodynamic equilibrium essentially often boils down to some type of maximization or minimization. So maximization of entropy or minimization of energy, you need to then sort of minimize the internal energy subject to the constraint of your pressure and temperature. And then therefore you have this Legendre transform in here, where you convert the internal energy into G, and then you look for the minimum of the free energy to find sort of the thermodynamic equilibrium state for such a system. And when you write this in a differential formulation, du is given as TdS minus PdV plus, and you have these chemical potentials if you have a mixture of components. And after this kind of transformation, you get this expression for the differential free energy or Gibbs free energy, and that looks like this here. Now, in a chemical experiment, when you hold pressure and temperature constant, obviously this expression here, then dt equals 0 and dp equals 0. So the free energy change under these conditions is only the sum that is shown here, sum over chemical potentials times the change of your particle numbers. That means that in a chemical reaction, what essentially changes your kind of free energy is a change or exchange of particle numbers or change of particle numbers. And you can see that these chemical potentials are related to the free energy via this expression. It's essentially the partial derivatives of the free energy with respect to the particle numbers at constant P and T. Now, why am I telling you this? So when you apply this whole kind of concept to chemical reactions, and we start with the simplest possible reaction, it looks like this here. So maybe one molecule A converts into another molecule B and vice versa. That means that every time you lose a molecule A, you gain one molecule of B. And this can be rewritten with these kind of differentials for the number changes. So sometimes you have like these coefficients in front of these when, for example, several molecules of A convert in one molecule of B or vice versa. And this is encoded in these so-called stoichiometric coefficients. And the convention is that the stoichiometric coefficients for the starting sort of reactants are counted negative, and those for the reactants or for the products are positive. So as a chemist, of course, you write something like 3A in equilibrium with 2B. That means that your stoichiometric coefficient is -3 in this case because you have the minus here. So just to explain that. Now, one thing that one can introduce is the so-called extent of the reaction. So that's kind of a reaction coordinate, psi, that tells you how far the reaction has gone from kind of left to the right, in a sense, in this equation. And this is obtained by just dividing this dn, this kind of dn by the corresponding stoichiometric coefficient. And that allows you to rewrite the chemical like this dg as the sum over chemical potentials times stoichiometric coefficient times this d psi, which is kind of the differential reaction coordinate. Now, when you read a chemistry or biochemistry book, you always read these delta G's for a reaction. And the delta G, that's the free energy or free enthalpy change of the reaction, is in principle defined by this quantity. So you now take the derivative of G with respect to this reaction coordinate. And that obviously just gives this sum over the chemical potentials times the stoichiometric coefficients. And in the case of our super simple reaction, that looks like this here. So the stoichiometric coefficient of A is -1 and of B it's +1. Therefore, the delta RG, so the reaction free energy, is minus mu A plus mu B. That's just the difference of the chemical potentials of the products and the starting the reactants. And then, of course, at equilibrium, you want that this equals 0. So at equilibrium, there's no driving force for the chemical reaction to go in one or the other direction. And therefore, the chemical equilibrium state, and of course, you can also go back and say that this corresponds to dg equals 0. But if the dg is 0, this is kind of at a minimum. Therefore, the equilibrium condition for a chemical reaction simply is that the chemical potentials of all your reactants must be the same. So this is what's written here. And now we come to some like the more useful things for our lecture. So now the chemical potentials are related to the concentrations of the reactants like this here. And this you can in principle motivate by looking at so in a textbook this would be derived by looking at the number of possibilities how you can arrange particles in the system. So that essentially is is an entropic factor in principle. So therefore, your chemical potential varies like this here, RT ln C concentration divided by some kind of reference concentration. And this is just the so-called standard chemical potential. And with this expression, you can rewrite the delta Rg like this here. So now you sum over these quantities, and you see that the first part of it, the sum over the mu I 0 times nu I, they give you some kind of standard value. That's the so-called standard free enthalpy of the reaction. And the rest of the expression gives you this here. And now we come back to the equilibrium thing. So at equilibrium, we have already said that this delta, the free energy change, must be 0. So you just solve this equation for delta Rg 0, and that tells you that the standard free energy change is related to the concentrations via this expression. And now you now the definition of this new of these coefficients comes in handy, because here you have a sum over logarithms. You can convert that into a logarithm of a product, and now this product actually is something that we call the equilibrium constant of the reaction, and the definition of the signs for these stoichiometric coefficients now actually takes care of which of the concentrations are in the numerator and which are in the denominator, and therefore the kind of final. So this is consistent with how you conventionally write your equilibrium constant. Now the equilibrium constant now is just the product of the concentrations normalized by some reference concentration to the power of your stoichiometric coefficient. And to make this clear, again, in this very simple example, we have these two molecules in equilibrium. Now, if we apply that kind of this definition here, you have Ca divided by reference concentration to the power of the stoichiometric coefficient. You have Cb divided by the reference concentration. In this case, the reference concentration cancels out, and you get the sort of definition of the equilibrium constant that you are probably used to. It's just the fraction, or you just divide the concentration of your products by the concentration of the starting products. And this is a very well-known relation, of course. It essentially, if you solve kind of this equation here for the equilibrium constant, you get this. And it essentially tells you that the equilibrium constant is is e to the minus delta, the standard free enthalpy change divided by RT. And now the important point is that this really must be the standard free energy. So this refers to this kind of standard state with a certain concentration, and you cannot just and this really refers to the chemical equilibrium state. So it's not exactly the same as just this is not the Boltzmann relation in a sense. It comes somehow from a different source. Now, when you have a more complicated reaction, and this is relevant for us in the context of DNA, because, for example, one DNA strand binds to another DNA strand and makes a DNA duplex, you have such an expression. Now, the free energy change of the reaction is given by the standard change and plus this expression that involves the concentration. Now, here I actually included this C0, which is the kind of the reference concentration. And of course, in most cases, you omit this expression. I'm just, it depends on the situation whether you actually want to write that or not. you see that the ln, of course, only likes to have an argument without any units. So this should be unit free. So in principle, if you plug in a k that looks like this here, you would have something with units. So that's actually not okay. And in reality, we would actually not really look at the concentration, or not in reality, but if you do it right, in principle, you always look at the concentrations normalized by some standard, or in chemistry, there's also the concept of an activity of a substance, and you would actually use activities here, which also are without any units. In any case, I think you get the point. So most of the case, we for example, just take C01 molar, and that just gives us this additional unit that we need. And this is connected to the definition of the standard state. So if the standard state refers to concentrations of 1 molar, then this is the concentration that you need to use here. So in many cases, we look at such a reaction from the opposite side, because it's often important to look at the degradation or dissociation of a complex into its parts, and then you actually look at the complex C in equilibrium with its dissociation products, and then you actually look at the inverse of this equilibrium constant, and this is the so-called KD, or the dissociation constant of the reaction. And you see that the KD now has units of a concentration. So this is a concentration times the concentration divided by 1. So 1 will remain. And so KDs are very often just used to quantify the strength of binding of something. And we will probably also come back to this later in more detail. So typical KDs for binding, for example, of let's say an antibody to its antigen is maybe a nanomolar. And that means that at 1 nanomolar concentration, half of the the antibody will be bound to your antigen, or something like that. So it gives you a measure sort of how at which concentration the binding partners will bind. So if you have concentrations that are much higher than the KD, you will actually saturate the interaction, so they will be bound to each other. When you are much below the KD, then there will be no binding. But I will come back to this in the lecture. Now, the standard conditions I already mentioned. In chemistry, now one takes standard pressure and temperature as usual, these quantities here. And in biochemistry, there's this additional condition that the concentration of water is taking the concentration of water. That's 55.5 molar, and the pH is 7. And that is actually specified because if you had, let's say, standard concentration like 1 molar, you would have a pH of 0, of course, and that would not be very biological. And also there are certain sort of salt concentrations are fixed. So that's not extremely important. I just wanted to mention it, that it actually matters, at least for a physical chemist, it would matter how you really write it down. So this delta Rg with that 0 here would refer to this state. And in biochemistry, you would in principle make like this additional mark here to indicate these biochemical standard conditions. But we usually ignore this. I just wanted to mention that this would be the correct way to do it. So finally, thermodynamics is somehow is connected to kinetics in that sense that if you look at such a chemical reaction, then there will be conversion of molecules A into B and vice versa. So when you're at the equilibrium state, the concentrations are not supposed to change anymore, and therefore any reaction in this direction must be compensated by another reaction in the opposite direction. Now, such a chemical reaction can be converted into a differential equation that looks like this here. So that this just says that the change of the concentration of A over time is given by the conversion of A into B that happens with that rate minus K plus times that concentration. But you also gain molecules of type A by the reverse reaction, that's plus K minus KB. Now at equilibrium, the concentration of A is supposed to stay constant, and then you can just equal kind of this equation to 0, and that you can solve, and it will tell you that the concentration of B divided by concentration of A at equilibrium is given by the ratio of these rate constants. But on the other hand, we also know that this ratio is given by the equilibrium constant, and this is how you usually convert kinetics into thermodynamics. So you can, for example, calculate the equilibrium constant from a delta G value, and then you know that the ratio of these two rates must be the same. One thing that is not clear from that equation is actually there's an additional factor that could actually cancel out. So you don't know the absolute time scale of these processes. You only know the ratio of these rates. So there must be, you would need to have an additional information that tells you how large at least one of these two rates actually is, so you can calculate the other rate from it. Otherwise, you only know the ratio of them. So that's essentially that. And okay, I'm soon done with all the formulas. That's just as a crash course for everybody. So there is actually a relation to statistical mechanics, and so the free energy can be obtained from the partition function in statistical mechanics, and that plays a role in that sense that I don't know, maybe some of you have this biophysics book, Physical Biology of the Cell by Rob Phillips et al., and they work a lot with partition function formulations because they think that this is kind of more natural for a physicist to think about. But on the other hand, I think I'm more used to this kind of chemical description somehow. And but they are related, of course. And just as a very brief remark, how these are connected, maybe you have had that already in statistical mechanics anyway. So in a canonical ensemble, the partition function looks like this here. You just sum over the Boltzmann factors of all the states in your system. So each state that is, the states are somehow numbered with that index I, have an energy epsilon I, and you just sum over all of these Boltzmann factors. That gives you the partition function. And that is very useful because you can generate all the relevant quantities by differentiation, essentially, from this partition function. So when you differentiate with respect to this parameter beta, which is 1 over kt, you get this sum, and this obviously is the mean energy, because sorry, it's not the mean energy, it's energy. Okay, it's this expression. So when you actually take the derivative of ln of the ln of the partition function, of course you first get 1 divided by partition function times this expression, and this expression now is this sum, and that means that now this is actually, this expression is the mean value of the energy, because this is somehow the normalization constant of this distribution. So therefore you can relate the internal energy to the mean energy, and this is given by the derivative of the ln of the partition function. Now the entropy is in the kind of in the definition by Boltzmann, is just given by Kb times the number of possibilities in principle. And that is related to the probability. When you assume that every possibility sort of is just equally likely, then this probability p is just 1 divided by omega, and that gives you that expression. Now in the Gibbs ensemble, you look at a quantity that is kind of the average entropy, and that's given in principle by this expression. Then here, when each state only has a probability of pi, so when they don't have the same probability, but different probabilities, then the mean of this entropy expression would then be given by this here. So you see that this is the probability to be in that state, and that is the kind of entropy kind of that state. So you're averaging over all the entropies, and that gives you kind of this expression. And you can now just take the Boltzmann expression for these probabilities pi and insert that into the entropy expression. And after some calculation, which I'm not going through in the details, you get the first term here will give you the free energy. And this here just is 1 because of normalization, and the rest is the kb times ln of the partition function. Now this looks like this here, and you can rearrange that equation, and it gives you minus kb ln partition function is u divided by t minus s. And that gives you the familiar expression in the end. When you multiply with t, you get minus kt ln c is u minus ts. And u minus ts is, of course, the Helmholtz free energy. So this is one of these thermodynamic potentials that we had in the beginning. So therefore, and because Helmholtz free energy is kind of useful in this context of these chemical systems that we are looking at, because this refers to systems that are held at a constant temperature. This is the way how the partition function is connected to a quantity like a free energy. And this is something that we probably need later on in the lecture. So just remember that f is minus kt ln partition function. And yeah, a few more things. It's often useful to remember that kt is in electron volts, it's 26 milli-electron volts, but it's also 4.1 times 10 to the minus 21 joule. 10 to the minus 21 is not very convenient, so you can actually split that up into newtons and meters. And in biology or biophysics, we always work with around piconewtons and at length scales of nanometers. So you see that 10 to the minus 12 times 10 to the minus 9 just gives 10 to the minus 21. And so it's often convenient just to remember that kT is 4 piconewtons times nanometers. But the chemists don't like to use kT, they use RT. And for this, it's useful to remember that RT at room temperature is 2.5 kilojoules per mole. And in this kind of other units, it's 0.6 kilocal per mole. Now, after this short overview of statistical mechanics, we are going to very briefly talk about molecular interactions. And the point is that now what we want to look at in the end is which molecular kind of forces hold DNA double helix together. And this in the end influences its thermal stability, which is then measured somehow in thermodynamic quantities. So of course, one of the fundamental interactions is charge-charge interaction. So just two charges attract or repel each other, and they have a distance dependence of 1 divided by r. Now in biology, we always work at in salt in salt solutions, and it turns out that under such conditions, actually, a charge is screened by other charges and also by dipoles and so on, and a screened Coulomb potential doesn't look like this, but it looks like this for a point charge, for a sphere with a radius a. And the most important point to notice here is that now we don't have an 1 divided by r distance dependence, but it's the distance dependence looks like this here, and there is a exponential decay of the interaction, which is given by this Debye-Hückel parameter. And we will come back to this in a separate chapter on polyelectrolyte behavior. But what is important to notice is that two charged objects in salt solution don't feel each other until they get into contact on the order of the Debye length, which is typically maybe on the order of a nanometer or smaller. That just means that two charges simply don't interact with each other in salt solution when they are far apart from each other. Therefore, this doesn't really play a role in our context. Then there are all these dipole interactions. I'm quickly going through that because it takes too long and it's not really super important. Just as a reminder, in principle, you can have a dipole that consists of two charges, plus and a minus charge, and this dipole can now interact with a fixed charge q, and this interaction has a distance dependence. It depends, of course, on the orientation of the dipole, and it goes like 1 divided by r squared. This is in a linear geometry, so this theta, this angle, becomes 0, so this cosine becomes 1, and then it looks like this here. If you have two dipoles, it's different. Then you have this famous expression where you have where the interaction decays by this with this r/3 dependence, but there is also this angle dependence, of course. And then there's this issue that maybe your two dipoles might fluctuate in orientation. And if you average such an expression over all orientations, you get a potential that decays with the power of r to the 6. That's sometimes called Keysom interaction. And this is important to remember because this plays a role for certain biophysics techniques, like, for example, FRET. So first, the resonance energy transfer has this 1 over r to the power of 6 dependence, which is related to this type of thing, because two fluorophores can be seen as like two dipoles. They fluctuate all the time, and they transfer energy to each other via such dipole-dipole interactions, and therefore you have this r to the power of 6. And then there's actually another possibility that you have a fixed dipole and you have a charge cloud that can be polarized. So this is characterized by polarizability alpha. And you get, again, an r to the power of 6 dependence for this type of interaction. And it also is possible that two polarizable charge clouds can interact with each other. So a random kind of fluctuation in one charge cloud will lead to a transient polarization, which then polarizes another charge cloud, and then they are coupled. And this also gives you an r to the power of 6 dependence. So all of them are r to the power of 6. And therefore, they are sometimes just taken together. All of these three r to the power of 6, they all together are called dispersion forces or van der Waals forces. And they give you some type of this behavior with some constant. And this actually plays a role when you look at such molecular interactions. So often you model molecular interactions with a Lennard-Jones potential, and the attractive part of the Lennard-Jones is actually this type of interaction. whereas the repulsive force here is typically modeled with something like this here. I'm sorry for skipping this kind of too fast, but I think that you probably have heard all of these things. Otherwise, it's more like a summary that one can look at if you want. Another thing that is important in biology are these hydrogen bonds, and these play an important role also in DNA. And they occur because we often have chemical bonds between something that is very electronegative and hydrogen. And these hydrogen bonds can be treated in a similar way as a covalent bond where two atoms sort of share, in a sense, an electron. And here they would share a hydrogen atom. And this can be described actually in a similar way as molecular orbitals with electrons. And so they have very different strengths for the different sort of possibilities that, let's say, when you have fluorine and hydrogen, these are extremely strong for hydrogen bonds, and they are weaker for the ones that are relevant for us in DNA. So in DNA, we have kind of these types of bonds. And now you can actually remember maybe how much energy we have in the thermal energy. So that was about 2.5 kilojoules per mole. So this is on the order of a few, maybe up to 10 kT in energy for such a bond. Yeah, so I'm skipping this. And now really the final part of all of our molecular forces that we need is the hydrophobic effect. That's something rather mysterious, which is kind of different from these kind of charge-charge interactions. And so the hydrophobic effect comes both from some enthalpic and some entropic contribution. And I copied this from a physical chemistry book just because I didn't want to try an explanation myself. But the idea is that you probably know that there is something like hydrophobic aggregation of molecules. So if two hydrophobic molecules are in water, they tend to aggregate. And the effect of that is that because water doesn't like to interact with these hydrophobic molecules, it needs to somehow make like some type of hydrogen bonded network around your molecule, your hydrophobic molecule. And now this has a certain surface, and a certain number of molecules are kind of bound in this kind of complex. Now, if these hydrophobic molecules come together, then they form a bigger aggregate that has in total a lower surface area than the individual molecules that are surrounded by these water kind of networks. And that means that water is actually sort of released by the aggregation of these hydrophobic molecules. So not so many water molecules are required to form that cage. And these water molecules, they gain in entropy because they now can move freely as they want. They are not bound there. And this is a major kind of entropic driving force for this process. And of course, also enthalpic contributions play a role maybe between these hydrophobic molecules themselves and also the hydrogen bonding of the water. And because it's a combination of these things, one cannot clearly say that it's not a real kind of, it's not only a molecular interaction in that sense, because it contains these entropic aspects. And now all these things actually play a role in DNA, and this is why I was talking about this. You see that when we now come back to our DNA double helix, finally, we have negative charges at the backbones. Then we have these molecules here that are these kind of planar pi electron systems. And so what plays a role is charge repulsion, for example, between the phosphates here or along the backbone. Everything is surrounded by salt solution. Therefore, this electrostatic charge is actually screened with this Debye-Hückel expression. Then there are hydrogen bonds here that you can see. These things tend to be hydrophobic. Therefore, there's a contribution of the hydrophobic effect in putting the bases inside of the helix, and they interact with neighboring bases via van der Waals forces, including induced dipole-dipole interactions and so on. So really, even though this was maybe a little boring, like with all these things, all of these forces actually play a role here, and therefore it's kind of really difficult to also calculate the stability of a DNA from first principles. There are heroic people in molecular dynamics that try to do ab initio modeling of DNA, where you essentially try to start with Schrödinger's equation and calculate how stable the DNA is. That's close to, well, it's very difficult, let's say. So other people then use a more heuristic approach where you parameterize the interactions between the protein, between the different molecules, where you have effective potentials that are known to work well to calculate interactions. Here in the physics, in the former physics, in the bioscience department, Martin Zacharias is performing these simulations, and they have a lot of experience sort of which kind of forces or force fields to use to get the desired results for these interactions. So a few more remarks maybe on the stability of the helix. So one thing that is important about the AT and the GC base pairs is that actually the distances within these base pairs are actually the same. That means that the distance from here to here is about 0.3 nanometers, but also the distance from this point to this point is the same as the the distance from here to here. And that is important in that sense that it gives the DNA a constant diameter. Because if there was a different distance, let's say from here to here for the AT base pair than for the GC, it would kind of change in diameter all the time. And this is one of the probable reasons why these base pairs are special, because in the end, an enzyme needs to be able to smoothly run over the DNA without much friction in a sense. And so this makes these two base pairs different from many other possible base pairs. And I think I already mentioned that last time, so that the bases can in principle make all kinds of base pairing, but the Watson-Crick base pairs are sort of special because of this property. But the hydrogen bonding doesn't really play a major role for the stability of the DNA helix. So this is really more for also for the replication process. It's important that the enzyme sort of the polymerase can check complementarity, and the base pair must exactly fit into the binding pocket. Otherwise, it will reject one of the bases when it is about to be incorporated during polymerization. So therefore, this plays a role in a sense of checking or making the binding specific, and it's not so much stabilizing the helix, even though, of course, this is often stated that AT is less stable than GC because you have two hydrogen bonds here and 3 here, but this is not entirely true. And there are major differences that depend on which of the base pairs is actually neighboring the base pair that you are looking at. And this shows that actually the so-called stacking interactions are actually the major stabilizing stabilizing kind of force in the DNA double helix. And this has to do with these, also with these forces that we have been just talking about. When you look at bases in the context of the double helix neighboring each other, then they have such arrangements. For example, adenine above adenine, adenine above cytosine, and so on. And in many cases, you see arrangements where, for example, there is one atom here in the center of one base, and there's an aromatic ring surrounding it. That's also here. It's not so clear everywhere, but this is often seen, at least. And this might can be explained that maybe this carbon at the corner has maybe some partial charge, and the pi system that consists of the electrons will interact with that somehow, and that has to do with such a kind of type of interaction. That's at least one contribution to this type of stacking, but in probably all of these things play a role somehow. So the also the pi systems, they can also interact with each other via dispersion forces, via sort of polarizing sort of each other. But there will be also permanent dipoles in these rings, because you have these heterozygotes with carbon and nitrogen, for example. And the carbon-nitrogen bond, of course, is kind of polar. So all of these things sort of play a role, and they play together in the exact arrangement of these bases on top of each other. Now, heuristically, people have characterized how the bases are exactly kind of oriented in the double helix using these parameters, which you don't have to remember. They sort of make sense, I guess, when you look at that. The idea is that maybe the base pairs, they could base pair via the hydrogen bonds, and they could shift like this with respect to each other. They could bend like this. They can make these twists and all kinds of things. They are all shown here. And they have these different names: shear, buckle, shift, tilt, stretch, propeller. And the DNA structure experts, they know all of these parameters. So I think that we are not really excessively talking about this, but there is a kind of biological role of DNA flexibility potentially. And so this is why I'm just briefly mentioning it here. So certain base pairs can kind of move with respect to each other more easily than others. And so some can kind of bend more easily. And there are certain DNA sequences that make more flexible double-stranded DNAs than other sequences. And there has been some speculation on the role of that. For example, that an enzyme sometimes needs to bend your DNA, and then it would be natural to assume that the corresponding sequences around the binding site are sequences that sort of allow the bending a little more. The other thing is that, for example, when DNA is wound around histones in the chromosomes, then they need to be bent very sharply, and that's a kind of common assumption that this preferentially happens at certain base pair steps that are particularly prone to be bent, or where the bases can shift with respect to each other. And therefore, there is kind of this suspicion that there is kind of a underlying code in the DNA that is not genetic, but that also encodes some type of mechanical properties that is necessary for binding to other molecules. And here's just one example that I think that we are not going, I don't go through these things here, but you see that this is about sort of moving bases in one or the other direction, or trying to make this propeller twist. And these stars, for example, they indicate a sterical clash. And the idea is, I think that when you, for example, move the upper base, you would shift it like this here, then it would collide with the molecule or the group of the base below, and also here and also here. And that means that in these situations, sort of the DNA base can only bend in one direction, not in the other, because they would clash. And that influences somehow the mechanics in the end. So for the last kind of few minutes, I'm finally talking about useful things, more useful things for you maybe. So how we usually look at DNA hybridization is the following. So there are two DNA strands that might be completely complementary. When they bind to each other, they make a double-stranded molecule. So in reality, this process probably happens like this, that the two molecules will touch each other and they will tentatively make one or two base pairs, and then the thing zips up after making this nucleation of the first helix. For short pieces of DNA, this process is supposed to be so fast that one effectively only looks at a two-state model, where you assume you cannot observe the intermediate. So this is just a two touch and then quickly form this duplex. Therefore, it's called two-state. And you only look at the duplex or the completely separated strands. And for this reaction, you can now write down our equilibrium constant as we discussed it. And this will be related to some delta G for the reaction that is shown here. And now one useful measure that people like to use to characterize stability of DNA duplex is the so-called melting temperature, Tm. And the idea is that when you heat up a double strand of DNA at a certain temperature, the two strands will fall apart and will become single-stranded. And this has to do with the fact that, like loosely speaking, your delta G, of course, is delta H minus T times delta S. And then you see that when you change T, you increase T, then you will change also this free energy, and you can therefore shift the equilibrium by changing this delta G through the temperature. So the melting temperature is defined as the temperature at which exactly half of your DNA is single-stranded, and you need to write down like a mass conservation equation here. So when you have a total concentration of DNA strands that we call C0, and this is just the concentration of strands that are strands of type A that are in single-stranded form, plus concentration of strands B that are in single-stranded form, plus 2 times the concentration of the complex, because there are 2 strands in one complex. So this is the total concentration of single strands in your solution. Now the melting temperature is defined like this. Now all the single strands have a total concentration of A plus B, and this must be half of the total concentration. So that's this equation. You can now actually, now I'm confused. Okay, you can insert that up here and bring that on the other side, then you get this equation that 2 times C is C0 divided by 2. That means at the melting temperature, actually the concentration of your duplexes is 1/4 of the total concentration of single strands. And now you can just calculate the equilibrium constant at the melting temperature. Yes?
Sorry, can I ask, what was the 2C?
The C is the concentration of the complex. So that means that, so that's just the total concentration, and some of the strands are either single-stranded or in the complex C, and therefore you have the factor of 2 here, because there are 2 strands in one duplex. And when you insert that into the equilibrium constant, now you get this expression, and that simplifies to this expression here. Now you can actually also, so here we now have the melting temperature in this Van 't Hoff expression. You can take the logarithm on both sides, and that will give you this expression here, minus ln 4 times psi divided by C0. Where did I define the psi? Psi is just, again, some reference concentration. You don't have to care about it. It's just a reference concentration to make this dimensionless. That just appears here. In biochemistry books, you will not see this psi because it's sort of understood that this is 1 molar. But as a physicist or mathematician, it hurts when you take the logarithm of something with a unit. Therefore, I have this in here. So that's, and then you can remember that the delta G is really delta H minus T times delta S. So the T cancels out and you get kind of this expression here. And from that, you can now actually solve for the Tm, and that gives you this expression. Tm is delta H divided by delta S plus R times Ln concentration divided by 4. You can do the same calculation for the case of a self-complementary sequence. So that's A plus A when something has kind of a palindromic sequence, so then it can bind to itself. And then the whole calculation goes slightly differently, and you don't have the factor of 4 in the end. So that gives you this expression. And I'm talking about these things because they have been really technically important, because what people did over many decades in biochemistry labs is now measuring melting curves. So what you essentially do, you have a double-strand DNA, and you heat your solution of the double-stranded DNA, and then you will see an absorbance signal that looks like this here. And there is an effect that is called hypochromicity that leads to a higher absorbance for single-stranded DNA than for double-stranded DNA. Therefore, when your DNA falls apart, your duplex falls apart, suddenly the absorbance will increase and you will have 2 separate strands. So the reason for this hypochromicity has to do with the fact that, of course, all optical transitions, they are always dipole-dipole kind of interactions. So you, in this case, when you illuminate DNA at 260 nanometers, that is where these pi electron systems absorb in DNA. And there are kind of these dipole transition, these how are they called? These matrix elements for the dipole transition matrix elements, and they somehow cancel each other up to a certain degree in the helix. So, and therefore the absorbance somehow, so when you vectorially add up these dipoles in the double helix, this is the effective dipole is smaller than when they are separated, and therefore the absorbance is actually smaller in the double helix than for the separated strands, and that's somehow the reason for that. In any case, you see this steep transition, and then you can actually note down the temperature at which this occurs, and you call that's the melting temperature. And now you go back to this expression or to this expression, and what you can then do is you just measure this for several concentrations of your DNA, and that gives you such a curve. Now you have several values for the melting temperature as a function of the concentration, and you can plot it like this with 1 over T versus the log of the concentration. And that ideally gives you a straight line. And from that, you can actually extract the delta H and the delta S. So from these melting temperatures, you can actually obtain the enthalpy and the entropy of the formation of the duplex. So now you have to do this for many different sequences. So, and this is what people did over decades for all kinds of sequences, concentration-dependent melting temperatures. You get many, many tables, and these tables are now the basis of standard thermodynamic prediction programs. So because I'm running out of time, I'll skip over that very quickly and come back to that later. But this is maybe also useful in case you want to do the tutorial. So all these measurements have been done, and in the end, well, what people found is that you can actually decompose any sequence into these so-called nearest neighbor parameters. And that is reflected by the fact that really the stacking between two neighboring base pairs plays a role. And this is a list of all possible neighboring base pairs. And here you have delta H and delta S values, and these have been sort of extracted from these measurements. So when you are given a certain sequence like this here, for example, you now decompose this sequence into these nearest neighbor parameters. And you see that these are nearest neighbors CG, GC, that's here, then GTCA, that's here, then TTAA, that's here. And you look up the corresponding delta H and delta S values from this table. Then you can calculate actually the total delta G for your helix. So that sort of assumes that there aren't any longer range interactions, that it's sufficient to only look at the nearest neighbors. There are additional correction factors for the initiation of the helix that has to do with the fact that when you make a helix, you lose entropy because the two molecules cannot freely move anymore. So you lose some entropy. Symmetry correction is more complicated. I will come back to that next time. It has to do with the number of states that a symmetric molecule can have with compared to a non-symmetric molecule. And then there is also penalties that have to do with whether you have an AT at the end or not. An AT tends to be destabilizing at the end. And it tends to be actually open. So if you have an AT base pair at the end of a helix, it's usually not closed but open. And all of these things sort of are in these tables. And all the thermodynamic programs that people use, and I don't have an example here. No. So in the tutorial, you're supposed to calculate delta G values using a program like NewPack, for example, that's online. And there you can just type in your sequence, and then these programs just take these tables essentially and calculate the delta G for you. In reality, it's more complicated, but this is essentially how it works. And when you have that from the delta G, you can then in principle also go back and calculate your melting temperature or other properties. So that maybe was like a long stretch. I think that we started with statistical physics and then talked about these molecular interactions, but they all really play a role in the context of DNA stability. But on a practical level, nobody really cares about these things because they are very complicated, and this is why things have been just experimentally parameterized. So all these programs work with experimentally obtained parameters, and I think that they are still being refined, but it's simply it's essentially not possible really to calculate such parameters from first principles. Therefore, it's kind of better to start to use the experimental things. And with this, I'm going to continue next time. I have to admit that I'm not here next week again. There will be tutorials on Wednesday, so somebody will do it for me, and then the problem is that the week after there is a holiday on Thursday. So, and because we are losing more and more, I will give an ordinary lecture on Wednesday in 2 weeks, and instead of a tutorial. So, and for the people who cannot attend on Wednesday, I'm sorry, but I'm going to record the lecture anyway. And hopefully later in the semester, we will have an ordinary schedule. So next week, only tutorial on Wednesday. I will send an email to everybody. Okay, thank you.